So the goals of our assay was basically to develop a fast, sensitive, and reliable and reproducible in vivo model for CRS. All right, so I'm going to talk to you today about predicting immunotoxicity for antibody drug candidates using a humanized mouse model. I should also tell you this platform also works for CAR T therapy. Well, so the goal of this assay when we started about 4 years ago is to develop a fast, sensitive, reliable, and reproducible in vivo model for cytokine release syndrome to help de-risk therapeutic antibody and CAR T development for preclinical development and hopefully moving these drugs into the clinic. We want to be able to differentiate individual antibodies for the same target, so like high affinity versus low affinity antibodies. Evaluate drug effects on individual PBMC donor, the variability of donor effect that we need to be able to capture. Looking at adverse drug combinations, combining a bispecific with a checkpoint inhibitor, what is that going to potentially do? And also to determine the drug dose response and with efficacy and safety in the same mouse. So the idea is to be able to compare many variables in the same mouse at the same time. So back in 2016, Germaldi went ahead and did an analysis of in vitro cytokine release type assays. And his analysis is on the left-hand side of the slide. And the right-hand side of the slide is our in vivo platform. And essentially what he saw with the in vitro platform was this, you got an inconsistent response across multiple types of assays, like whole blood versus isolated PBMCs. It's dependent on the test article's mechanism of action, and there's evidence of false positives and also false negatives. These in vitro assays don't offer a dynamic range to discriminate low, medium, or high responders. It's a little harder to do high affinity and low affinity antibodies. Quantification of the cytokine levels varies dramatically between the different platforms and different labs. There's some beautiful studies coming out of NIBSC where they looked at same antibodies in like over a dozen different laboratories in pharmaceuticals and biotechs, and they saw dramatically different effects of the antibodies in these in vitro assays for CRS. So it's highly variable. And also, it lacks physiological conditions to offer in vivo relevance. Our approach was basically to tackle this last question of in vivo relevance. And so we have a holistic physiological model to be able to look at this humanized mouse. So we can look at the human immune system, but also the downstream effects on the actual mouse, on its organs, on its overall health as well. I'll talk to you more about that in a minute. This model allows you to graft human tumors, okay, into these mice to be able to look at toxicity and efficacy in the same platform at the same time, obviously. We get reproducible results with, you use different types of test articles. We've done this with OKT3, T-Genero 1412, anti-CD28, bispecific antibodies, and again, CAR Ts. And we also have a large bank of leukopak PBMCs that have all been pre-characterized for positive and negative controls for CRS. So this is the basic model. Okay, it's a 6-day model. And I can tell you that this model can range from anywhere from 6 days to 16, depending on your therapeutic and what kind of endpoints you're looking for. But it's a very rapid model. At day 0, you radiate the mice and inject your PBMCs. At day 5, you confirm humanization, and I should tell you that every mouse is injected goes on study. You get a very, very, very high humanization rate. At day 6, you add your therapeutic, and then you do your readouts. We use multiple mouse models. We use the NSG or the SGM3 or the MHC class 1, class 2 knockout to ask different questions. Again, 6-day assay, we use between 500,000 to 30 million PBMCs. We usually use between 500,000, okay, or between 5 million and 30 million PBMCs. Usually use between 5 and 15 million PBMCs in our platform. We only use 4 to 5 mice per arm. The data is very tight. You'll see the error bars in all the data. Once again, we run a bunch of different therapeutics, OKT3, anti-CD28, T-Genero, bispecifics and drug combinations, CAR T, autologous and allogeneic as well. And once again, our endpoints are things like cytokine release, mouse body temperature, mouse clinical evaluation. Okay, what happens to the mouse? Downstream organ toxicities in the mouse, effects on immunophenotyping, and once again, CRS in relationship to efficacy. Now, all this can be done in the same mouse with only 4 to 5 mice per group. So once again, we can see these are different PBMC models shown above. Depending on the mouse model, okay, we can see T cells, B cells, NK cells, and variable levels of myeloid cells. For cytokines, we see the Th1, Th2 T cell panel, but we also have a myeloid panel where we pick up things like MIP1 alpha, RANTES, IL-12P40, IP10, and so on. We also can detect granzyme A, B, and Fas ligand. So depending on the mouse model, depending on what you're trying to accomplish, we can get a very diverse set of cytokines and cytotoxic endpoints as well. So we're talking about a systemic response to immunotherapy. Okay, and the key here is going into your laboratory model or into people. The whole goal is, of course, to get efficacy, be able to shrink your tumor or decrease tumor burden. But because of that, this often leads to a cascade of events that are not desired. Some of that is cytokine response, be very, very high in some individuals, lead to cytokine release syndrome. You see changes in the immune cell profile, such as T cells. So right after treating with bispecifics or different therapies, you can see changes in the CD3, CD4s, or CD8 populations. I'll show you some of that data. You also see some downstream effects like body temperature changes in the mice, the organ inflammation and toxicities. I'll show you some of that data as well as death in some of these mice under some of these conditions with these therapeutics. We can capture all that in this platform. This is an example of T-Genero 1412. Okay, Adrienne mentioned this in the beginning of the talk. This is back in 2006. Essentially, this is a drug. It's an activator of T cells that was past everything. It passed the in vitro assay. It was treatments in rats, in monkeys. It showed that the drug was safe to go forward into a phase 1. It showed no toxicities at all. And unfortunately, 30 minutes after giving T-Genero 1412 by IV to 6 healthy individuals, all started to get fever, and they all ended up getting organ toxicities downstream. It was a severe reaction. The assays back in 2016 were unable to detect T-Genero 1412. As you can see here in this experiment, our assay is able to detect it. We also can get a dose range with the drug itself. We have picograms per mil on the y-axis, and we have time on the x-axis. And you can see 1 hour, 2 hours, 4 hours, and 6 hours. PBS in black, anti-CD28 is in green, and the 3 different doses of T-Genero 1412 are shown from pink to purple. And as you can see, over time with with this therapeutic, IL-6 is peaking around 4 hours post-treatment. TNF on the right-hand side is one of the first cytokines to appear with bispecifics or T-cell engagers or T-cell antibodies. And you can see that 1 hour, we have about 600 picograms per mil of TNF, and by 6 hours, it drops down to almost basal levels. So essentially, we're able to detect human cytokines in this platform with T-Genero 1412, and it's just a proof of concept about the value of the platform. So our model, we believe, shows the clinical hallmarks of CRS. It captures a large number of them. We're able to see effects on liver enzyme levels can go up. We can see changes in the immune cell infiltration. We see caspase-3 or cells going through apoptosis and infiltrates, and we can see hepatocyte necrosis occurring as well. This experiment, looking at the histology of the organs, was essentially a 10-day assay, where at day 0, we put the PBMCs in, at day 6, we treated, and then at day 10, 4 days later, we harvested the organs, and we did the histology. So this is showing abnormalities in the liver of these mice. This is comparing PBS to anti-CD28, H&E staining, caspase-3 staining, and human staining for human CD45 levels. And as you can see, for the H&E staining, the PBS in the top left, on the bottom left, you can see the high levels of human immune cells infiltrating the liver of these mice 4 days post-treatment, single dose. On the far right, if you look at human CD45s, you see the large numbers of human CD45s immune cells that are in these anti-CD28 treated livers of these mice. In the center panels is caspase-3 staining. As you can see, a lot of these immune cells are actually going through apoptosis and dying within the liver itself. Here is looking at for single cell necrosis. Okay, this is H&E staining, and the yellow arrows are pointing to some of these essentially single cell necrosis or hepatocytes going through the cell, essentially dying. And as you can see, with either anti-CD28 or T-1412, these hepatocytes are dying. You are getting liver toxicity, and it should also tell you you get elevated liver enzymes in peripheral blood as well. This is long. We got PBS, anti-CD28, T-1412. Then we have H&E staining. Below that's caspase-3, and below that is the human CD45s. And from H&E staining and the staining for human CD45 cells, you can see there's a huge amount of infiltration, okay, in these mouse lungs with the human immune cells. Okay, and on the right-hand side is the histopath scoring, where we're essentially you have grades 0 through 4. And with both anti-CD28 and T-1412, all 5 mice on study treatment, you see you get grade 4 histopath in the lungs of these mice compared to the PBS control. Antibody comparison and selection. Okay, essentially the idea here is, can you differentiate different affinity antibodies for your drug development platform? In this experiment, we use PBS, group 1, OKT3, and anti-CD28. Keytruda is our negative control. Keytruda obviously is approved drug from Merck. It's known not to cause CRS, and then we use it as a negative control. Then we have high affinity and low affinity antibodies targeting BCMA, so the BCMA CD3 bispecifics. And we use 3 different doses for the high affinity, 0.7, 2, and 6 mg per kg. For the low affinity, we actually went higher. We used 2, 6, and 18. And we wanted to evaluate these drugs for cytokine levels of CRS. So this is showing 2 different cytokines being released, interferon gamma on the left and IL-10 on the right, picograms per mil on the y-axis, and the different therapeutics on the x-axis. You got PBS from left to right, PBS, OKT3, anti-CD28, Keytruda. Then you have your 3 doses of your high affinity CD3, BCMA CD3 bispecific. And then in the blue, okay, you have your low affinity. And the first thing you can see is that with the high affinity BCMA CD3, we're getting high levels of interferon gamma being secreted, as well as high levels of IL-10 in comparison to Keytruda or your low affinity BCMA CD3 bispecific. So one thing you can see right now is that with the higher affinity, you get higher levels of cytokine release, and you can differentiate the low versus the high. One of the effects you see of these single treatments of this BiTE is the fact that you have a significant effect on the T cell population at 24 hours post-treatment. OKT3, which is the orange on the far left, essentially it targets the CD8 population, and you see a dramatic drop in CD8s, in CD8 populations. Okay, anti-CD28 or Keytruda don't really affect that population, but the high affinity BCMA CD3 in purple, pink to purple, you see a dose response when it comes to loss in the cell population. And you don't see any significant loss with the low affinity antibody. In the CD8s or CD4s that are remaining present in these 24 hours, you can see with the CD69 positive cell populations, an increase in activated CD4 cells in this slide with increasing doses. So we have a nice dose response, and we see a nice consistent data when it comes to differentiating high versus low affinity antibodies for cytokine release and also immunophenotypic effects. This is showing a mouse clinical score. Okay, how's the health of the animal? And this scoring goes from 0 to 4. Essentially, all mice must be taken down at a score of 3. And this is just showing that this is at 6 hours post-treatment, so a very quick response. You can see with all the mice with OKT3 reach a score of 1. And also you can see a nice dose range, dose response on the health of the animal with the high affinity antibody, but not the low affinity. So once again, you can see some effects on the mouse at a very quick time point within 6 hours of your treatment of the mice. Drug combinations. So a lot of research is being done combining bispecifics with checkpoint inhibitors as an example. And we've done a lot of them with solid tumors as well as liquid tumors. And I'm just going to show you one example. This is in a high tumor burden model where we use the MDA-MB231 cell line. In this case, we luciferase tagged it, so we want to be able to follow the tumor burden as well as looking at cytokine release. So we're looking at the burden, the tumor itself, but also the cytokines released after treatment with the EGFR CD3 bispecific. Okay, plus or minus a anti-PD-1 drug. So in this experiment, we did day 0. We added our PBMCs. We waited a little longer to go ahead and confirm the humanization and of the mouse, and also to get the baseline xenogen imaging of the tumor cells. The next day, we added a therapeutic, and we did imaging for the next couple of days. This is cytokine release, okay? And this is, once again, this is with Keytruda by itself, EGFR CD3 by itself, and a combination in red. We look at IL-6, TNF, and IL-2. And what you can see is that with EGFR CD3, which is in purple, with IL-6, you're getting about 300 picograms per mil, 400 picograms per mil, excuse me. Keytruda, once again, around 300. The combination with IL-6 is giving you about 900 to 1,000 picograms per mil. For TNF, the results are even more dramatic. The EGFR CD3, purple, blue combination in red, and IL-2, once again, a very dramatic response when you combine these two together. Now, I should tell you this is in this donor. Other donors you see a different effect, and I'll show you some more data about that later on. But the point is there's donor variability on how these therapeutics combine, and there's something to take a note when you're going forward with your research program to make sure you look at enough PBMC donors to make sure you understand the combination effect when it comes to cytokine release. This is just with interferon gamma and IL-10. This is showing on the left-hand side your TNF levels, and the right-hand side is your tumor burden. So once again, your combination is in red. You're seeing about 800 picograms per mil of TNF. Okay, and this is at 6 hours post-treatment. On the right is the xenogen imaging. So at day 10 is before dosing. You can see your PBS, your EGFR, BITE, your Keytruda, and your combination are all about the same level. Day 14, which is post-treatment, single dose, you can see that essentially the EGFR CD3 by itself or in combination with Keytruda pretty much eliminated a large part of the tumor burden, and then it's probably about a 95% or higher loss of tumor cells in these mice. From this experiment, one thing you can conclude is that the combination didn't give you any additional benefit when it comes to efficacy, but it gave you a was detrimental when it comes to cytokine release. Excuse me. Reproducibility. So the whole point of finding this platform is to have reproducible results from experiment to experiment. And so this is an experiment that's done the second place. I'm sorry, I apologize. This is an experiment done with donors 9636. It was done 2 months apart. Experiment 1, experiment 2. And we're using different passages of tumor cells, and we're also, in this case, it's Raji. We're also looking at different dilutions done at different days, 2 months apart. And this is showing the left-hand side is picograms per mil. On the right-hand side is fold increase. And you're seeing these, this is the raw numbers going forward. This is showing you the effects of basically these 2 experiments done 2 months apart. 9636 experiment number 1 in orange and experiment number 2 in blue. And you can see once again picograms per mil on the left-hand side. Not exactly the same numbers, but the same trend, similar results. If you look at folds over background, you see that many of the dilutions superimpose on top of each other. So it's we like to think this platform has good reproducibility, even 2 months apart in 2 different experiments. Okay, what are the effects of CRS in a patient population? This is an experiment we did early in our process. This is 9 these are 9 different normal PBMC donors treated in black is PBS, OKT3 in orange, and anti-CD28 in green. And you can see the significant variability in interferon gamma levels of IL-10 in this patient population. In our paper we published last year in FASIP, donor 213, the 4th from the right, was done in 5 different experiments, and that data is shown to confirm reproducibility of the data we're getting. But as you can see, there's significant variability in how people, different normal PBMC or different individuals, respond to these therapies. Even more dramatic is with the BCMA CD3 bispecific I showed you before. This is 8 different donors. The high affinity is in purple, and the low affinity BCMA CD3 is in blue. And as you can see with 0585 and 8058, you can get a 20-fold increase in interferon gamma and IL-10 with treatment with this therapeutic, 6 hours after treatment. While other donors, you see essentially minimal effects or very low increases in cytokines over background. So you can see the need to be able to look at a good number of PBMC donors, I better understand your potential cytokine release issues, possible toxicities that you might eventually reach when you get into the a phase 1 trial. So how would you use this CRS platform? You can do early preclinical and late preclinical. For early preclinical, you can de-risk your pipeline for ranking different antibodies or molecules. You only really need 1 or 2 PBMC donors, only 5 mice per group. The length of the study is 6 to 10 days. In this readout, you can get a standard cytokine release, and you can compare with efficacy. So efficacy and cytokine release at the same time. You only need 15 mice for the whole study. And once again, if you add more compounds, it's only 5 more mice per group for each group that you add. For late preclinical, you're looking at 4 to 6 donors. Still 5 mice per group. The study can be, essentially 6 to 10 days. Standard cytokine analysis, organ tox, paired with efficacy. Talk about maybe 60 mice from the total study, and each donor has 15 more mice. So to summarize, my apologies, I'm finding a cold, obviously. JAX has developed a fast, sensitive, and reliable and reproducible in vivo model for CRS, help you de-risk your therapeutic antibody for preclinical drug development. You can do efficacy and cytokine release of the same mouse. You can evaluate the individual responses of individual donors. You can give reproducible data. You can differentiate different antibodies based on affinity for the same target. You can look at drug combinations, and you can look at downstream toxicities in the liver and lungs by INC, as well as liver enzyme levels. And here are our collaborators at NIBSC, GSK, University of Massachusetts. Many shows, of course, at Bar Harbor and my laboratory in product development. Thank you very much.